The second step in phloem translocation is the translocation of photosynthate from source to the sink. It is a long distance transport. In angiosperm, this process is best explained by a model known as pressure flow model, which was first proposed in 1926 by German physiologist Ernst Munk. Based on this model, he proposed a hypothesis known as pressure flow hypothesis. According to pressure flow hypothesis, the transport of photosynthate from source to the sink is driven by a osmotically generated pressure gradient between the source and the sink. The pressure gradient is established as a consequence of phloem loading at the source and unloading at the sink. First, let us discuss about this model. Then we will see that how the pressure flow hypothesis is applicable in case of phloem translocation. In this model, there are two osmometers, osmometer A and B. Osmometer A has a concentrated sugar solution and B has a dilute sugar solution. Both are covered by the semi-permeable membrane, which allow only water molecules to move in and move out. These two osmometers are connected by the tube T, and this whole setup is immersed in a water bath. Because the sugar solution in osmometer A is concentrated, due to this, osmosis will take place and water molecules will diffuse into the osmometer A. As a result, hydrostatic pressure will develop in the osmometer A. This hydrostatic pressure will help in the flow of some of the sugar solution from osmometer A to the osmometer B through the tube T. This process of flow of sugar solution from the osmometer A to the osmometer B will continue until an equilibrium is reached. If we can add sugar to the osmometer A and remove sugar from the osmometer B, then the process will go on continuously. According to pressure flow hypothesis, a similar process also takes place in the plant during phloem translocation. According to Munk, a similar system also operates in the plant in order to drive the process of translocation. In plant, the sieve tube through which the translocation process occurs is comparable to the tube T. The xylem and the surrounding apoplast is similar to the water bath, and the source cells that include mesophyll cells where photosynthesis takes place is comparable to the osmometer A, and the sink organ, that is the root, is comparable to the osmometer B. So according to pressure flow hypothesis, the translocation of photosynthate from source to the sink is driven by a osmotically generated pressure gradient between the source and the sink, and this pressure gradient is the consequence of phloem loading at the source and unloading at the sink. According to pressure flow hypothesis, the translocation process is a passive process. It is driven by the osmotically generated pressure gradient between the source and the sink. Now let us see how pressure flow hypothesis is applicable in case of phloem translocation. So this is the sieve tube, which is composed of a number of sieve tube elements, and these are the sieve plate, which is porous. And nearby sieve tube, this is the source cell or the mesophyll cell where photosynthesis takes place. In between the source cell and the sieve tube element, this is the companion cell, which also is essential in phloem translocation. And toward the end of the sieve tube, this is the sink cell, for example, root. And nearby, this is the xylem vessel. This is the source end of the sieve tube, and this is the sink end. At the source end, energy-driven phloem loading takes place. So there is accumulation of sucrose in the sieve tube element. This accumulation leads to a decrease in the solute potential of the phloem cell. So the water potential also decrease because the concentration of the sugar solution increase. In response to this, water molecules diffuse from surrounding vessels and other apoplasts into the sieve tube element. At the source, energy-driven phloem loading takes place with the help of H plus sucrose symporter. As a result, there is accumulation of sucrose in the sieve tube element at the source end. This accumulation of sucrose causes a decrease in the solute potential or osmotic potential. As a result, the water potential also decreases. In response to that, water molecules from the surrounding vessels and other apoplasts move into the sieve tube element at the source end. As a result of this diffusion of water molecule into the sieve tube element at the source end, a pressure develops. The hydrostatic pressure develops, whereas at the sink, phloem unloading occurs. Whereas at the sink, phloem unloading takes place. As a result, the concentration of sucrose decrease in the sieve tube element at the sink end. As a result, the solute potential increase. So, the water potential also increase. Whereas at the sink, phloem unloading takes place. Because of phloem unloading, sucrose and other sugar molecules leave the sieve tube element and move into the sink cells. So, the concentration of sucrose and other sugar molecules in the sieve tube element decrease. As a result, the solute potential increase. So, the water potential also increase in the sieve tube element at the sink end. When the water potential in the sieve tube element at the sink become more than the water potential of the xylem, then water molecules move out from sieve tube element to the vessels. As a result, the pressure decrease in the sieve tube element at the sink. So there is a pressure gradient. That means the pressure is greater at the source end of the sieve tube and the pressure is minimum in the sieve tube element near the sink. So there is a pressure gradient. So there is a pressure gradient. That means the torsion pressure in the sieve tube element near the source is higher, whereas the torsion pressure in the sieve tube element near the sink is lower. This difference in torsion pressure is generated by osmosis. This osmotically generated pressure gradient is the driving force for the pressure flow of phloem sap through the sieve tube. This is all about pressure flow hypothesis. It is the widely accepted hypothesis explaining the translocation of photosynthate from source to the sink.